And we're good. Okay, so again, this chapter is chapter 11 from our textbook. It is not going to be on the exam, but what you'll find is this process of meiosis is very similar in many ways to our process of mitosis. And a lot of what we're going to do with our time today is going to be relevant as you work your way through chapter 10. So first question we need to deal with, what are the products of meiosis? We already know what the products of mitosis are, right? The products of mitosis are two genetically identical nuclei. And if mitosis and cytokinesis both happen, your products are two genetically identical daughter cells. That is not what the products of meiosis are. Okay? And so this figure that comes out of a really good textbook, not yours, not that yours isn't a good textbook, but the figures from your textbook, when I put them on slides, don't look good. So I usually pull my figures from another text. But this does a really good job of demonstrating the products of meiosis. So when you look all the way at the right of this figure, it tells you what you're left with after this process is over. And we are not left with 2 cells, but rather 4. And we are not left with genetically identical cells. We can tell that because these chromosomes are color-coded. Okay? And so even if you struggle to see the specific color, you should be able to see the differences. And we have 4 genetically distinct cells. And not just any 4 genetically distinct cells, but cells that have half as much DNA as they ought to. Right? Typically, a cell has 2 full copies of the genome, like this. This is a typical cell in G1. Here is that cell following S phase, when the DNA has been duplicated. And then we get to the end of meiosis, and what we have are cells that have 1 copy of the genome. Okay? So the products of meiosis are 4 genetically distinct haploid cells. Haploid meaning having 1 copy of the genome, as opposed to diploid, which means having 2 copies of the genome. Okay? And so that's what you need to keep in mind when you're working through meiosis and what needs to happen. You need to remember the products of meiosis. Just like when we were working through mitosis and cytokinesis, you needed to keep in mind that after S phase, we have twice as much DNA as we ought to. And we have the 2 genetically identical copies of every chromosome are physically attached together. We need to fix that during the process. Well, if you can remember that this is what you need to produce, you should be able to reason through what needs to happen during meiosis. Sum this up in text. Meiosis generates haploid cells that can be used in fertilization. So in sexual reproduction, you're going to take one of your cells, it's going to fuse with a cell from another individual to generate a normal embryo. And normal embryonic cells have 2 copies of the genome. So if you took a cell from your body that has 2 copies of the genome and fused it with a cell from another person that had 2 copies of the genome, now you have a cell that has 4 copies of the genome. Bless you. That's not good, right? So this process of meiosis generates cells that have a single copy so that when they fuse together, 2 cells with a single copy, you have a cell that has 2 copies of the genome. One that came from one parent and one that came from the other parent. Now these cells are only made in the gonads. And in males, they are spermatozoa. And then those spermatozoa will mature, or in some organisms we call them spermatids, and they will mature into sperm. These are formed in the male gonads, the testes, and then ova, the female gametes, are made in ovaries. And so this process is called gametogenesis. Gametogenesis is the process of forming gametes. The mechanism of gametogenesis is meiosis. Okay? So gametogenesis is the process of forming gametes. The mechanism of that is meiosis. Now, to put this into context, human gametes have 23 chromosomes, okay? Which should make sense because a typical cell in our body has 23 chromosomal pairs, Okay? Unless you're male, and then you have 22 chromosomal pairs and one partial pair. Okay? Where you have two unique chromosomes, you have a Y chromosome and an X chromosome, but there are some regions of those that pair up, that form what we call complementary regions. And so male gametes have 23 total chromosomes, one copy of the genome. Now, meiosis does a couple of things. One, it just facilitates fertilization in general without getting a situation where you have way too much genetic information. But it also does a really good job of increasing overall variability. Because any one gamete you produce will almost always be genetically distinct from another gamete that you produce. It is virtually impossible for you to produce two genetically identical gametes. It's virtually impossible. It is technically possible, but it is virtually impossible. So meiosis produces 4 daughter cells from a single germ cell. And so what that means is in spermatogenesis, when we are generating spermatids or spermatozoa, and we take one germ cell, we get 4 spermatids that can mature into 4 sperm. And in oogenesis, where you are generating female gametes, you take one germ cell and you generate 4 cells. But what do we know about the ovum? Do any of you know the main difference between an ovum and a sperm? The most obvious difference? Size. It's huge. When meiosis happens to generate ova, the cytoplasm is not distributed evenly. And so the ovum ends up being really big. The human ovum is almost large enough to see with the naked eye. I think it's about 0.3 millimeters in diameter, which some people can actually see it, most people cannot. It's just too small to see with the naked eye. Okay? So it's a big cell. Very, very large cell. All right, anyways, moving on. Next question. What is recombination and when does it happen? So there's something that happens during meiosis that makes it virtually impossible to produce two genetically identical gametes. So in this image, you see Prophase I. Where have we seen prophase before? Mitosis. Seen it during mitosis. And what are some of the things that happens during prophase? The chromosomes condense, the nucleus dissolves. We form the mitotic spindle, or we begin forming the mitotic spindle. Those centrosomes migrate to opposite poles. Okay? During Prophase I, in meiosis, something really important happens, and that's your two copies of the same chromosome associate together in a very, very important way. And so what this is showing you here is our two versions of, let's say this is chromosome 1. It could be any chromosome, but we'll say chromosome 1. And so you see we've got two versions of chromosome 1, and something is happening right here. Something's happening right here. A portion of the maternal chromosome is actually getting in and interacting with the paternal version of chromosome 1, and vice versa. And this is called crossing over. When you actually get the two versions of chromosome 1 interacting in such a way. It's called crossing over or recombination. And so in recombination, specifically, so crossing over is just this association, the formation of this unique structure, and we'll talk more about that in a minute. In crossing over, you actually mix those two chromosomes. So at the end of this, you don't have a maternal and a paternal version of this chromosome. You actually have two versions that are both maternal and paternal. They've exchanged information. And so at the site at which they exchange information, they are physically attached together. And this is really interesting because in mitosis, we were talking about that following S phase, the two sister chromatids are physically attached together, right? They're still attached together following S phase before meiosis. And then on top of that, our two versions of chromosome 1 are now physically attached together. So you actually have 4 copies of that chromosome that are all physically attached together, right? Because each chromosome now, your paternal chromosome is actually 2 copies of that chromosome attached together after S phase, right? And your maternal version of that chromosome is 2 copies of that maternal chromosome attached together after S phase. And now the maternal chromosome and the paternal chromosome are physically attached together. 4 copies of that chromosome physically attached together. You're like, wow, this is weird. Something unique is happening. And so to sum this up, what recombination is and when it happens, well, just a little bit of information. So recombination is going to increase variability even further because it creates combinations that didn't exist before. So when you make gametes, you will put chromosomes into those gametes that have never existed before, because they are a combination of your mom's chromosome 1 and your dad's chromosome 1. So the chromosome 1 you put into your gametes, it's a unique version of chromosome 1 that's never existed before. And it's the same thing is true for chromosome 2 and chromosome 3 and following. Even with the X and the Y, because there are portions of the X and the Y chromosome that associate together and will cross over. Now there are unique portions of chromosome Y that when expressed develop maleness in mammals, and then in birds the opposite is true. Females are heterogametic, have 2 different sex chromosomes. But there are even regions of that X and the Y chromosome that will cross over and create combinations that have never existed before. And so that's what recombination does. And so one sister chromatid from each pair crosses over. And so this forms a structure called a chiasma, plural would be chiasmata, and forms what we call a tetrad, where instead of having 2 strands of DNA that are attached together, you actually, in these regions, have 4 strands of DNA that are all attached together at these chiasmata, forming what we call a tetrad. Now, these happen as a result of actually bringing those 2 copies of chromosome 1 into physical contact with one another. Bless you. Bless you. And so recombination happens, to answer the question of when does it happen, chiasmata happens, or recombination happens when the chiasmata disappear during prophase I. That's when recombination happens. So what recombination is it's a mixing of the paternal and the maternal version of each chromosome. Okay? So if you're going to look at and focus on chromosome 1, recombination would be a mixing of the maternal chromosome 1 with the paternal chromosome 1 to produce a copy of chromosome 1 that is neither maternal nor paternal. It's both. Yeah?
Which chromosomes are recombined?
All of them.
Oh, really?
Yep, recombination happens at least once on every chromosome every time you go through meiosis. And even on our smallest chromosomes, usually happens at least a handful of times on the chromosome. That's a good question. Yeah?
So it mixes the maternal and paternal?
Yes.
So it's neither or?
Or you could look at it as the chromosomes you are putting in. So your chromosome 1 that you're putting into your gametes, you could either say it's neither maternal or paternal, or you could say it's both. I mean, both of those things are true. It doesn't look exactly like your mom's version of chromosome 1 that came from her ovum, and it doesn't look exactly like your dad's version of chromosome 1 that came from the sperm. It's a combination of the two. Yeah, it's a combination of the two. It's a unique version of chromosome 1 that's never existed before, which is kind of cool, right? That every time you make a gamete, you are building chromosomes that have never existed before.
And
Yeah.
what happens in the tetrads?
Oh, the tetrads are just at that point at which they're crossing over. You have 4 strands of DNA that are all attached together. It looks a little bit like this. So typically 2 strands of DNA would look like this, right?
Yeah.
Sorry, I know the green is hard to see, but that's what color I have. So you've got 2 strands that are attached together with hydrogen bonds. In these tetrads, you actually have, wow, that is really bad. see if this green is any better. I just need to carry a black expo marker around. There we go. Oh, that's a lot better. Okay, so here's what DNA typically looks like, with two strands that are attached together by hydrogen bonds. At the point, at these chiasmata, I'll call this a chiasma because there's only one there. It looks like this. You actually have 4 strands of DNA that are all associated together. And we have a special name for these tetrads. Do you want to know what it is? It's kind of bonus information, but it's kind of cool. It's called a holiday junction. It's because it was named by a British scientist because mom and dad are on holiday together. Is that cool? Oh man, that is so cool. I think it's cool. My next door neighbor is from New Zealand, and we got back from a trip, and she said, "Oh, you're back from holiday." I said, "No, there's no holiday. We went on a trip, went on vacation." Yeah.
So what's the difference between a chiasmata and a tetrad?
Nothing.
Oh, okay.
Nothing. Yeah. Yeah, a chiasmata, a tetrad, a holiday junction, they're 3 names for the same structure. Yeah. 3 names for the same structure. That's a good question. All right, this is lecture break time. And so what I want you to do is this. I want you to think about where we're at right now, where we have both versions of chromosome 1 are physically attached together. Okay? Both versions of chromosome 2, and I'm not talking about the sister chromatids, I'm talking about your maternal version with 2 sister chromatids is physically attached to your paternal chromosome with its 2 sister chromatids. Okay? So that's where we're at right now. And I want you to basically spend a little bit of time, maybe only a minute or so, talking with those around you. And I want you to determine how is that going to be fixed and what is that actually going to accomplish. So how do we make it to where they are no longer attached together, right? Because that can't persist forever. How do we get it to where they are no longer attached together? Chromosome 1 from your dad is no longer attached to chromosome 1 from your mom, okay, with their now exchange of information. And then when they are detached, so I want you to, figure out when, not necessarily when, but how that's going to happen. And when they are detached, what does that actually accomplish? What have you accomplished after you separate them? All right? Take about a minute, maybe 2. We'll see how these conversations are going, starting now.
Okay, so they're attached together
There's a. What does it accomplish?
Well, that sounds good.
I have to review that. That does cutting. Okay. So, I mean, unless there's some other structure, that's all that I know right now that actually cuts. So that's what I was.
We'll have fragments from different aspects to the variety.
Yeah.
So that if it goes through like same processes, it's processes. Anaphase and all that. Meiosis, and then whatever you say. If they do that, then like they're gonna pull apart the mixed ones, right? The pictures of fragments. Yeah, right. And then that's gonna do. That adds variety, doesn't it?
Yeah, it does have. You're right. Something that
All right. Okay. How are we going to separate these 2? Do we have a mechanism, I don't know, let's say from chapter 10 at our disposal that does a fine job of separating chromosomes? Yeah, Autumn.
Anaphase?
We do. We've got anaphase, and what is it that's actually separate? What's doing the separation directly? The spindle fibers, specifically directly, we call them the kinetochore microtubules, right? They grab hold of the sister chromatids. during prometaphase, they align them into the middle during metaphase, and they pull them apart during anaphase. And then at the same time, non-kinetochore microtubules are elongating the cell, also functioning to separate the sister chromatids. Man, we have a wonderful mechanism. Why not do that to separate the two versions of chromosome 1? Take kinetochore microtubules, but now instead of having one from one pole grab one chromatid, one from the other pole grab the other chromatid, how about have one from one pole grab the maternal chromosome, one from the other pole grab the paternal chromosome, and now when you pull them apart, you don't pull apart the chromatids, you pull apart the two versions of chromosome 1. And so the paternal version ends up at one pole, the maternal version ends up at the other. But you know, by this point, crossing over has happened, and it's not truly a paternal chromosome anymore. It's actually part paternal and part maternal. But anyways, one copy of chromosome 1 ends up at one pole, and one ends up at the other. And then if this coincides with cytokinesis, you end up with one ends up in one cell, one ends up in another. And so now we've taken our two copies of chromosome 1, let's say, and you have put them into separate cells. Which means the second part of our question, what have you accomplished? What have you accomplished in that? Well, you've accomplished some variety, absolutely. Anything else?
Crossing over.
Yep, crossing over has increased that variety even further. But we've just cut the DNA in half. Because remember, at the beginning of mitosis, we have all 4 copies of chromosome 1, right? Because you have 2 copies of chromosome 1, but they've each been duplicated. So you have 4 copies of chromosome 1. They all line up in the middle during metaphase, 46 chromosomes long. Do you remember talking about that last week? Yes? On Friday? Maybe on Wednesday. So during mitosis, during metaphase of mitosis, we have a line 46 chromosomes long that contains all 4 copies of our genome. Now after this, after we separate those copies of chromosome 1 and the copies of chromosome 2 and the copies of chromosome 3, we have 2 cells that each only have 23 chromosomes. Now each of those 23 chromosomes is still 2 sister chromatids physically attached together, right? But we've halved the DNA. We've cut it in half. Once we separate our 2 versions of chromosome 1, each cell only has 1 copy of it. Now that 1 copy, again, it's actually 2 copies physically attached together, but it's 1 copy instead of 2. Well, it's 2 copies of 1 copy. Yeah, Davies.
Does that mean we've already gone through meiosis?
We've gone through the first half of it. At that point, the first half of meiosis is done, and you've already accomplished halving the DNA. You've cut the DNA in half. But we still have an issue that each version of our chromosome is basically two sister chromatids. And so we've still got to fix that problem. Because if that doesn't change, you end up at the end, you still have 2 copies of the chromosome, right? They're just physically attached together. That doesn't seem normal, and it's not. So that has to be fixed as well, which we'll talk about in just a moment. And you'll love it. No questions, thoughts, concerns? You're like, man, that's so wonderful. Dr. Ingold, now I understand a little bit better what we were talking about last week. I'm excited for Friday. Just going to destroy this exam that you put in front of me? I hope so. I hope so. Believe it or not, I do not write exams with the intent to make you do poorly. Also, believe it or not, I do not get too upset when you perform poorly, right? So I don't know if you feel guilty when you're like, man, I just really didn't do well. Dr. Engel's probably really sad. I've been doing this a long time. I've learned to not allow my emotions to be driven by student performance on exams. I have not yet, though, learned how to not allow my emotions to be driven by the Dodgers' success. But they had a good weekend, so I feel great. I feel great. Gee, Russell Martin came in and made his pitching debut. I don't know if you know who he is or if you even care. But he's been in Major League Baseball for, I think, 14 years now and played a whole bunch of years in the minor leagues. And he made his pitching debut for the Dodgers. It was beautiful. Beautiful. You should go look it up. Anyways. All right. So, I didn't even read the question. What is the process of meiosis and which one is similar to mitosis? So the thing about meiosis is meiosis happens under meiosis 1 and meiosis 2. You actually have 2 steps, which is how you can take one cell and generate 4, right? Because 1 times 2 is not 4. 1 times 2 is 2, but 2 times 2 is 4. Yeah.
I have a quick question about is it chiasmata?
Chiasmata.
In the textbook, it showed the 2 chromosomes in the X shape, like sandwiched together.
Mhm.
But is this the individual strands of DNA?
Yes, yeah.
Okay.
Yeah, so this is not like the entire chromosomes. So this would be, yeah, we've got like, a whole version of this chromosome, and this one's like down here, and then this is like here. I don't know if this is helping you, if this is making it worse, but you've got, and this is actually only in one of the places, because then you'd have, oh gosh, this is going to get really crazy. But I'm going to do it. I'm doing it anyways. So here's our other chromosome, because remember these are 2 sister chromatids attached together. Here's where they're attached together. Okay, the 2 sister chromatids. Each sister chromatid is a double-stranded DNA, right? That's what DNA is. It's double-stranded. It's a double helix. And you're like, Dr. Ian, why don't you draw this as a helix? Because I can't. Oh yeah, and then this one, oh man. Yeah, then we just, right up through our chemistry homework. There's our others. And so here's a sister chromatid, here's a sister chromatid, here's a sister chromatid, here's a sister chromatid. But at one point, at the chiasma, those 2 sister chromatids in the middle are physically attached together, forming this tetrad, where all 4 strands of DNA are associated.
So does it uncoil?
Yes. Yep. Yeah. There are machines that ensure that this happens. It doesn't just happen accidentally. Because, the chromosomes are heavily condensed at this point, and you should not be able to access like a specific strand, right? Unless you have a machine that will loosen them at a region and facilitate this happening. And we have those. And what's interesting is some people groups, those machines are more active than others. So in some people groups, their recombination rate is higher than in other people groups, which means that the amount of variation in their offspring should be higher. You're like, that's so cool. And in what kind of people groups is that? And it's people groups that have spent a lot of their time in a very variable environment. Okay. But anyways, that's another story for another time. Okay, so meiosis happens over meiosis 1 and meiosis 2, so you'll see things like this. Prophase I. Okay, you already saw this. We showed prophase I. What's happening here? Crossing over. We've got an association between our 2 copies of chromosome 1. You see our nuclear envelope is dissolving, our spindle fibers are forming, our chromosomes are condensing. Here we have prometaphase I. Our kinetochore microtubules have made contact, but look at this. One of them's contacting this chromosome 1, one of them's contacting that chromosome 1. There's nothing attached to this sister chromatid. You see that? There's nothing attached to this sister chromatid. Okay? There's prometaphase I. Here's metaphase I. They're lined up at the middle of the cell, but now notice they're not lined up single file. They're lined up side by side, that the maternal version of chromosome 1 is lined up next to the paternal version. But look at that, they've exchanged some information. You see that? You see that? They've exchanged some information. And then here's anaphase I, which that's our mechanism, okay? Those kinetochore microtubules shorten, the non-kinetochore microtubules stretch the cell out, and you end up with your 2 versions of each chromosome ending up at opposite poles. Levi and then Chris.
So you've got these 2 chromosomes that have exchanged some information, but
Yes.
are they identical?
Not anymore. And so now, so like during mitosis, our 2 sister chromatids, they're identical to each other. After crossing over, the 2 sister chromatids, they're no longer identical to one another. You see that?
Yeah.
Because this one, the way it's drawn, it's all maternal. This one is like 80% maternal, 20% paternal. Okay, and then here's 80% paternal, 20% maternal, and then 100% paternal. So after crossing over, the sister chromatids are no longer genetically identical. Yeah.
I'm cell division after fertilization? Is this what we're looking at now, or is this?
This is prior to fertilization. This is in the process of making gametes.
Okay.
Yep. No, you're fine. There's no need to apologize. Yeah. This is in the process of making gametes. So in males, this is spermatogenesis. In females, this is called oogenesis. But or just generally gametogenesis, right? The process of making gametes. The mechanism is meiosis. Chris.
So normally in mitosis,
Yes.
all 4 of those chromosomes would actually be lined up.
Yes, they would be lined up single file, 4 chromosomes long.
Gotcha.
But during Metaphase I, they're lined up side by side. Okay. And what's kind of cool here, when you look at this, so it's showing you them detached at this point, but they're actually still physically attached together. At the point where there used to be chiasmata, they're still attached there, not as chiasmata, but they're still physically attached there. It's almost like they're holding hands. And it's so cute because it's like the maternal and the paternal, and they're just, I mean, they're not on holiday anymore because there's no holiday junctions. They've come back, but their relationship's doing better now, right? Holding hands, it's so beautiful. It's so beautiful.
So
Yeah.
is this the point where it goes from diploid to haploid then?
Yes, at this point it goes from diploid to haploid. But only if this is accompanied by cytokinesis, because this is all still one cell at this point.
Okay.
But here's the next one, Telophase I and what we call interkinesis, the cytokinesis in between meiosis I and meiosis II. You see, you've got these microfilaments forming that contractile ring. You see that? We saw that in cytokinesis before. Going to pinch these cells into 2. And yeah, at this point now we have 2 haploid cells.
Okay.
Now those haploid cells have unusual chromosomes, in that the chromosomes have 2 sister chromatids, but they are haploid cells. Okay, and now meiosis I is done. Yay! And the whole process is halfway done. Yeah.
So for this image, we have just 2.
Yeah. So you started with 4, and now we have 2. We have half as much DNA.
Yeah, but in a real cell.
So you'd have 23. Yep. You started with 46, and then after this you would have 23 in each cell. Yes. Now each of those 23 chromosomes would have 2 sister chromatids following this, but yes, you'd have 23. But that's a lot of chromosomes to fit into an image. Yeah. But it does it in your cells, you know.
The cells become haploid. Does that mean they develop certain characteristics that are kind of resistant to certain diseases and stuff?
Potentially, but I mean a gamete is technically not even living.
Oh.
You know, like a gamete is a cell that facilitates sexual reproduction, but a gamete by itself, you could argue, is not even a living cell. It's not capable of metabolism. It's not capable of responding really to all stimuli. There are some stimuli that they can respond to. Like the sperm communicate a little bit with ova, and then the sperm respond to chemical cues to know which direction to move in. You know, so it doesn't metabolize. It can't make copies of itself. I mean, so you could argue that a gamete is not even a living cell. Now, it facilitates sexual reproduction in living organisms, but an individual gamete, it's not a normal cell, and it can't be because it only has 1 copy of the genome. You'd have a major expression issue with some of our genes that you need 2 copies of to get proper levels of expression. And you only have 1 copy of those now. Well, actually, you still kind of have 2, but they're attached together. Yeah.
So do gametes, like, do they read the DNA that they have to do certain things, or are they just
Not really. It's already got to most of it has to already be in place. So yeah, there is some degree of gene expression happening in gametes, but very little. Most of it has to already be in place, that they actually have to inherit the machinery necessary to carry out whatever their function is. Yeah. And that's part of the maturation process when spermatid mature into sperm. You know, part of that is providing the machinery necessary for them to do their work, which is basically just a ton of mitochondria and a ton of ATP and a ton of sugar to metabolize. So, because the sperm, I mean, they've got to swim like crazy. And they've got to swim a long distance. I mean, it's not that great of a distance, but when you're the size of a sperm cell, it's a huge distance. So, nothing? Okay. All right. So now meiosis I is done. Now what you're going to see is prophase II, prometaphase II, metaphase II. And notice, we still have to have the normal things happen during prophase I. We've got to dissolve the nucleus. We've got to recondense the chromosomes. We've got to rebuild these, the spindle. And then in prophase II, now our kinetochore microtubules have made contact, but now look, there's one attached to this chromatid and one attached to that chromatid. Now we're starting to see something like what we saw in mitosis. So the question here is, how does meiosis work, and which step is basically identical to mitosis? Well, we know meiosis I isn't, because it had a lot of differences, but mitosis II, it's looking pretty good, look pretty promising. And then here's metaphase II. Our chromosomes are lined up single file now. Here's anaphase II. Our sister chromatids are pulled apart like they are during anaphase of mitosis. And then here's telophase, where we decondense the chromosomes, rebuild the nuclear envelope, degrade the mitotic spindle or the meiotic spindle. And so meiosis II is virtually identical to mitosis. And again, meiosis II, virtually identical to mitosis. What's the only difference? The genetic material is a little bit different, and the amount of it is different. Is that what you were going to say, Levi? You have 23 chromosomes instead of 46. Okay? So you have 23 chromosomes lined up single file instead of 46 chromosomes lined up single file during mitosis.
And
Davies.
and you produce 4 daughter cells instead of 2.
Yes, but now from each daughter cell, we're going to produce 2. So like this started as a single cell, and it's going to end up as 2, just like what happens during mitosis. The difference is at the beginning of meiosis II, we're starting with 2 cells instead of just 1. And each one of those cells will be split into 2 cells. All right? So here's another little figure that just puts both meiosis I and meiosis II into a picture. And so here's meiosis I. Oh, and this compares meiosis II. I don't know why this is saying prophase I, metaphase I, anaphase I, when this is all meiosis II. That's really confusing and wrong. Sorry. But this is showing it side by side with mitosis. So here's metaphase of mitosis. Oh no, this is metaphase I. But why did it say meiosis II at the top? Oh, wow, this is really... Oh, is it... What is happening? Meiosis II is all down here. Wow, this diagram is not helpful at all. Let's do this. I'm going to delete that slide and pretend nothing ever happened. You are with me. At no point did I ever show you that slide. You have no idea what that slide was, or... Wow, that was a terrible slide. Oh, well, what are you going to do? Okay. So I want to take a minute to just remind you of where we are. We're not going to go to the next framing question because we don't quite have enough time to finish it. So I just want to real quick take a couple of minutes and just summarize where we're at and give us a foundation for picking up on Wednesday. Okay. So you need to remember, the goal of mitosis is to generate 2 genetically identical nuclei, right? And the success of any given cell is determined by its ability to generate 2 genetically identical nuclei. So that when cytokinesis happens, you get 2 genetically identical cells. That determines the success of any given cell going through mitosis. Because mitosis is used for growth, right? We were talking about this last week. It's the primary way in which multicellular organisms grow is through mitosis. But you don't want to generate a line of cells that are genetically distinct, okay? Genetically unique. Because then that creates kind of a mosaic situation, and it makes it to where your immune system actually has 2 different individuals it has to recognize as you, right? That's not good, right? That's where you start getting some weird things happening. And it's actually one of the main causes of autoimmune diseases. When you actually do end up with mitosis that doesn't work properly, and you end up with mosaicism. We have 2 genetically distinct cell lineages, and you're basically your immune system recognizes one of them as you and the other one as foreign. Okay, and you run into autoimmune issues. Anyways, the goal of meiosis completely different. The goal of meiosis isn't necessarily to generate genetically distinct cells. That happens during the process. The goal of meiosis is to generate haploid cells. That's the goal of meiosis, because that's what needs to happen in order to make sexual reproduction possible. Otherwise, the only way to reproduce would be by mitosis, right? You just bud off a new individual that's going to be genetically identical to yourself, which is cool if you're like destroying life, right? You're just like rocking it, you know, you've nailed this environment. The reality is environments change. So the environment you're adapted for is not going to be the environment that your offspring face, which provides some value to be able to generate offspring that are genetically distinct from you. So that's a byproduct of it, but the goal of meiosis is to generate haploid cells. Now, in that process, we do generate genetically distinct cells. Okay? It's always going to happen that way. And on Wednesday, what we're going to do is we're going to take some time and we're going to look at how do these processes, how does crossing over and how do some other processes that happen during meiosis generate diversity? And again, that's not necessarily a goal, although that's the reason why sexual reproduction exists. Or actually, the reason sexual reproduction exists is that's how it was designed to exist. But the purpose of it and the value of it is to make every generation able to endure an environment that is dynamic and is constantly changing and fluctuating. All right? So the goals of mitosis and meiosis completely different. So there's something in the mechanisms that are completely different, and all of those differences are found in meiosis I. Okay? Where your two versions of every chromosome associate together. Okay? That doesn't happen during mitosis. And so that the first step, the first thing you pull apart isn't sister chromatids. The first thing you pull apart are your two versions of each chromosome, because they've associated together in a way that they don't during mitosis, but they do during meiosis, during Prophase I, as they cross over. All right? Have a wonderful early afternoon. Those of you that are in lab, I'll see you in a bit. Those of you that are in lab on Thursday, I won't see you in a bit. I'll see you on Wednesday.